Welcome to the EMEA Hemodialysis Spotlight webinars. Today we will talk about the membrane perspective of uremic toxins, which ones should or can be removed. My name is Peter Stammenkel. I'm a professor at the Karolinska Institutet in Stockholm, Sweden. And I'd like to thank you, all healthcare workers, as you continue to provide world-class care to patients with chronic kidney disease. We have many attendees today, and all lines have been muted as a courtesy. We do, however, want you to engage with us during the program. In the user panel, please feel free to submit your comments and questions at any time during the program. We have also included handouts of both the presentation and literature article, which will be discussed today. And if you have any additional questions after the webinar, please send an email to the respective region seen on this slide. We have a comprehensive webinar schedule, as you see here. Over the course of 8 webinars, we will discuss articles from the supplement recently published in the Clinical Kidney Journal entitled The Duality of Dialysis Membranes, the Attributes and Ramifications. With these webinars, we aim to help facilitate a better understanding of the functioning principles of hemodialysis, with a focus on the critical role of dialysis membranes. Be sure to register to attend when you receive the announcement via email. We will share upcoming dates later in the program. Our panelist speaker for today is Dr. Xia Tao. Dr. Tao currently leads the clinical and laboratory research team at the Renal Research Institute in New York City. Dr. Tao received her Bachelor of Medicine from Guangxi Medical University in China and a PhD in Biomedical Engineering from the University of Massachusetts. She has a clinical background combined with research experience in the fields of drug discovery, medical device design and validation, and clinical research. And she also has an expertise in the development of therapies for better removal of uremic toxins. And with this, I would like to turn this webinar over to Dr. Tao. Please.
Thank you, Professor Stevenson. Good morning, good afternoon, and good evening, everyone. My name is Xia Tao. I am from Renal Research Institute. It's really my pleasure here today to present the work on behalf of the co-authors on this article we recently published on the Clinical Kidney Journal. So this is an article to address the question that among the uremic toxins that known to us, which ones should be removed most desperately, and what can we do with them with current hemodialysis technology, therapy strategies. In today's talk, we will analyze this topic from the dialysis membrane perspective using an evidence-based approach. In some parts of the presentation, external reference will be included to provide further insight into the discussion. So here's the disclosure of the program. Before we dive into answering the question I just mentioned, I think we should take some time to discuss what uremic toxins really are, as the definition has been evolving over the past decade. Simply speaking, uremic toxins are a subset of solute that found accumulated in kidney disease. This group of accumulated solute are called uremic retained solute, as you can see a list on the left-hand side. There are some features associated with them. They usually have elevated levels in kidney disease population, especially kidney failure, because of the declined kidney clearance or increased production. Their level oftentimes found increased along the progression of kidney failure, and their levels are oftentimes quite different among individuals. So among those solutes, some has been studied more frequently and believed to be linked with damages to biological systems related to uremic symptoms. This solute could be generally called uremic toxins. However, I believe the level of evidence supporting their toxicity might be strong in some, while relatively weak with the others. So we will talk about more of this point in the later part of the talk. On this slide, I am summarizing the relationship of uremic retained solute and uremic toxins for you. So laboratory analytical methods has been advanced greatly in the recent years. But not all the solute in biological fluids has been identified or quantitated accurately. Among those we can detect, there are some found elevated in kidney disease comparing to normal population. In those retained solutes, uremic toxins are the ones that has been studied and linked with negative biological effects. So uremic toxins originate from different sources, as you can see listed on this graphic. And then they are further metabolized into substance with different physiochemical characteristics. Traditionally, they were classified into three groups. One is protein-bound uremic toxin. One is water-soluble small molecule toxins. And then the other one is metamolecules. So historically, molecules with molecular weight larger than 500 was considered as metamolecule toxins. And the newer classification, they were broken down into more granular levels per their molecular weight. And the cutoff to distinguish protein bound or not was not clearly defined in the past. I think in the most recent classification, a cutoff of 80% binding was introduced to define if they are protein bound or not. So rather than just relying on physiochemical characteristics for classification, a new concept of classifying them based on their impacts on biological systems. In another word, their toxicity has been proposed. This indicates a trend in the research area, which has been also moving towards evidence-based approach. This is a trend that aligned with the approach we presented in our article. I do want to put some efforts and focus on this particular group. As you can see on the left-hand side on the source column, it's called gut-derived uremic toxin. It has been quite a hot topic in the research field recently. So gut-derived uremic toxins come from proteins, primarily from red meat. Some comes from fish consumptions. These proteins were broken down into amino acids, such as you can see in the figure, tryptophan, lysine, etc. They served as the precursors to be further metabolized by both human enzymes and microbiotas. Eventually, they are turned into uremic toxins in the gut. Their precursor are generated in the gut, and they finally metabolize into uremic toxin, sometimes in the liver, by adding a phosphate and other groups. So vegetarian diet has lower content of this type of amino acid, so they contribute less to the source for production. Meanwhile, vegetarian diets also offer prebiotics effect from non-digestible fibers. So in the recent years, there are a good amount of randomized controlled trials showing probiotics, prebiotics, and some symbiotics therapies can help to significantly reduce gut-derived uremic toxins levels in patients. So other than uremic toxin that generate from gut, there are also other toxins I want to mention. They are the production of normal biological functions. As you can see, some example listed in this table. There's quite some famous ones such as beta-2 microglobulin, uric acid. You will see their names appears again and again in the later part of this talk. So this comes to my favorite part of the talk. Some uremic toxins has relatively high affinity binding to albumin. As you know, albumin is a cargo that carries all sorts of substance in human blood. Although some larger molecules like amyloid betas found in Alzheimer's patients' body can also bind to albumin in blood, protein-bound uremic toxins are usually referred to solute smaller than 500 molecular weight in the field of nephrology research. So as you can see on the right-hand side, I show a figure which is the structure of 3D structure of the albumin. There are many binding sites has been identified and marked on this figure. So different substances have their own preference of where to bind to. Some has multiple binding sites. For example, free fatty acids has more than 7 binding sites has been identified. Some paper even suggest 9 more binding sites can be located. Some of those binding sites even overlap with where uremic toxin binds to. In most literatures, the binding sites are 3A and 2A, as you can see circled in red on this figure. They are also called sublow drug binding site 1 and 2. They are the primary binding sites for uremic toxins. But I do want to mention not only uremic toxins, other substances such as tryptophan, ibuprofen, they all bind to albumin. They primarily bind to drug binding site number 2. And other drugs like warfarin, furosemide, they like to bind to a different binding site number 1. So as they share the same binding site with uremic toxins, increased protein-bound uremic toxins levels in dialysis patients could contribute to alternated pharmacokinetics in the patients. So the relationship between albumin and protein-bound uremic toxins and maybe other substance as well in the blood. It's like those small fish, they swimming along with big fish like whales, sharks, and albumin being the big fish. Uh, uh, in this case, a whale, they attracts, they carries, and they release those small molecules in a very dynamic way. So toxins could hop on to albumin molecules and stay for a relatively short period of time, and they may leave. Once they return, they bind to another albumin, or maybe they bind to the same albumin. So this process goes on and on. So between those available binding sites, uremic toxin could bind to, they also have their own preference on which side they like to spend more time on. And this site is usually called their primary binding site. And they come with different binding affinity to albumin. In a physiochemical perspective, the higher affinity they have, the longer time they spend with this molecule. So as a result, the free fraction of those toxins in the blood, which is the portion that can be filtered by the dialysis membrane, also varies. So, for example, some toxins, as you can see on the first row of this table, CMPF, it's a toxin with such high affinity that almost no free fraction can be easily measured in plasma. So although these toxins are very sticky to albumin, they are still relatively weak if you compare to molecules like antibody, who theoretically resident on their ligands forever. So there is still a good chance to mobilize a protein-bound uremic toxin off albumin with some physiochemical methods. Now, I want to mention that as you can see listed on this table, a large portion of known protein-bound uremic toxins are gut-derived. But some are not. For example, canuric acid, it's a mammalian cell-derived toxin. And also, not all the gut-derived uremic toxins are protein-bound. For example, the TMAO, the one listed on the bottom of this table, it's completely water-soluble. It's one derived from consumption of fish. So besides protein binding, uremic toxin also come with different molecular weight. I think this is no surprise to anybody. But molecular weight plus protein binding create a broad spectrum of so-called effective molecular weight or effective size from the dialysis membrane's perspective. That's the size the dialysis membrane really sees. So in the later part of this talk, we would use the effective size of the molecules to analyze their interaction with dialysis membranes. So to summarize the first part of my talk, if you happen to miss any slides, so uremic toxins are solute that found first elevated in kidney disease patient. Second, they are studied and linked with negative biological effect. As I showed in the earlier slides, there's a long list of toxin. And also this list has been evolving with the advancement, development of lab analytical technology. In the recent year with development of omics technology, there will be more and more substance can be identified and detected in biological fluid. So we would expect this list to grow. But at the same time, there are also a growing effort to collect evidence to support all those toxins we found does mean something in terms of their clinical relevance. So the results of clinical relevance also impact how researchers perceive the toxins list. So in the from the membrane's perspective, the most relevant classification of toxins is by their effective size, which divides those toxins into three groups: small molecules, water-soluble toxins, protein-bound uremic toxins, and metamolecules. So now we know a little bit more about what uremic toxins are, how things been evolving, what their features are. But come back to the question, what we really trying to deal with, it's what to do with these toxins, right? And so we have better patient outcomes. Then we need to know first what hemodialysis can do currently. At the same time, such a long list of toxins show as shown earlier might seem to be quite overwhelming. So it would be good to know where to start with such a long list. So once we know these answers, we may have better idea which therapies should be used to handle those toxins. Now, I want to introduce this concept we proposed, called evidence-linked uremic toxin elimination approach. This is the concept we published in this article on CKJ. Simply speaking, with this concept, we aim to identify appropriate membranes or treatment strategies for those toxins has been shown with clinical relevance because of their toxicity. So the first step for us is to find out who are the true toxins, not just a marker. So as mentioned earlier, the levels of evidence vary among current recognized solutes. I would say only small portion of currently identified uremic retained solute has been studied intensively and present with strong evidence. Most available evidence limited to in vitro or animal studies. I would say at a clinical level, most those studies are observational studies. Some are even inconclusive, and studies that directly links with lowered uremic toxin levels to improved patient outcomes. Those studies are rare. I think probably because the contribution of single toxin is very hard to be dissected from the entire uremic milieu. So those pose obstacles for us to further clarify and understand the toxicity, the clinical relevance of individual toxins. So in this situation, what's the answer to this question? Which are the toxins with strongest toxicity? I think many attempts has been made to seek for the answer. I would recommend this article published by Professor Van Holder. As many of you know, he's a well-recognized figure in the field of uremic toxin research. I particularly like this article because it shift through all the current literature. I mean, by 2018 when the paper was published. And this paper evaluated more than 70 toxins across lots of biological systems and they developed a scoring system. So this provides a relatively objective way to evaluate the toxins. So far, I think this is the most comprehensive review we can find on this topic. And this article is still referenced in the most recent publication on CJSON for new classification of uremic toxins. So our analysis will be based on the findings of this article. So what did they find in this article? So the left-hand side table has summarized those toxins has been identified with the highest overall evidence levels. So this article, they look at different type of evidence. If a toxin come with lots of study with positive result, then it will get a score 1 to 4. And if there are a big portion of the study that did not deliver a positive result, then the score will be lowered. So with such a balanced scoring system, there are quite some toxins has been identified and ranked by their evidence levels. But those toxins you may be able to see, they are usually the toxins appears in research studies more often than others, than those who are not listed here. In another word, maybe they are those we are more confident that they are more likely to be toxic rather than those we barely know anything about, right? So interestingly, there are lots of water-soluble and middle-molecule solids were evaluated and reviewed and even listed in the candidate toxin list. But relatively fewer of them were present with strong evidence of toxicity. But in contrast to a protein uremic toxin, although the list of protein-bound uremic toxin is more, but those eventually get onto this list, they are relatively more than water-soluble and middle molecules solids. So one of the reasons I can think of may help explain this observation could be that most water-soluble solids, they are completely filterable. So as you can see in this figure, we published on the article, we compare the molecular weight of toxins that have been reviewed, and also compare them to this molecular weight and severe coefficient on both low flux membrane and high flux membrane. As you can see on those listed on the close to the left hand side in the red bracket, those are water soluble toxins. They have a severe coefficient equals to or close to 1 on high flux membrane, which is the blue line on this figure. And this portion of toxins compose almost half of the toxin that has been evaluated. So the large amount of toxins that has been identified, they could already been removed efficiently with current hemodialysis technology. So on the left-hand side of the figure, there are some molecules, they were removed to a varying extent. They are the molecules that with molecular weight larger than 10,000. So the priority of their removal should be examined by how confident the evidence are on their toxicity, in my opinion. So we will take a closer look of those toxins in the next slide. This is a similar figure as the previous one, but you can tell that in this case, those filterable ones, it's no longer in discussion. But if you take a look at this one, those between completely filterable and very filterable toxins, there are some middle molecules has been listed. The top two, interleukin 8 and beta 2 microglobulin, they are considered small middle molecules. Their severe coefficient it's not that far away from one. So they are quite filterable. That brings the question if it really worth the effort to enhance their removal to severe coefficient 1, because the offer this may not offer too much returns, especially when translating the dialyzer removal into reducing plasma levels in the body. And then those are highlighted in the red circles. If you look into Van Holder's review, they are those not considered with highest evidence levels in terms of their toxicity. So for those who are on the right hand side of the barely filterable cutoff, they are most protein bound toxins, because they form this albumin toxin complex. They have an efficient molecular weight larger than albumin, so they are barely filterable, and their removal cannot be achieved just by simply manipulating the pore size alone. So we would take a closer look to each group in the next slide. And I saw again the metamolecules, which are the ones that are removed by high flux membrane with a varying extent. I would say beta 2 microglobulin is the textbook example, as the one that has most established causality link to clinical outcomes. Its accumulation led to dialysis related amyloidosis symptoms, but it has been become a vanishing complication after high flux membrane was introduced. Now, so it's quite removable from high flux membrane, but for other metamolecules, as you can see, I cited a table from this publication published on 2017. By then, I would say most the evidence still limited to inconclusive or observational studies. So it's difficult to study metamolecules, because they are not simply a waste. Gut-derived toxins, Uremic toxins, they may be more likely waste, because they eventually go into kidney to be cleared, and their precursor comes out from feces. But metamolecules are a different story. They are usually functional molecules like hormones or cytokines. Sometimes it's hard to tell whether they are truly unwanted molecules, or they are elevated as a marker due to upregulation of biological response to kidney disease. So now we come to the part where current hemodialysis technology really have problem with removing the protein-bound uremic toxins. To be clear, the dialyzer membrane do remove part of these toxins, but only the free fraction of them, and primarily through diffusion. But unfortunately, this free fraction is very small in blood. For example, for a typical well-known uremic toxin, indoxyl sulfate and precursor sulfate, they only have 5 to 15% fraction of the total concentration is free in the plasma. And usually at the end of the treatment, you can even see this free fraction drops. We did some in-house work simulating the removal of those protein-bound uremic toxins with different treatment therapies. So the blue line, you can see that's a conventional hemodialysis. So no matter for example, for induxyl sulfate, the one on the left-hand side figure, it only has some reduction ratio like 30, maybe some cases 40%. Even on the hemodiafiltration, there is only a slight improvement. Hemodiafiltration could dilute the blood in a large extent, and that would sometimes shift the bound solute into free fraction. So this in this case could be more free fraction available to be removed, and the convection also contribute very minorly for the removal. And also, if we really increase the dialysis flow, which is the line in purple, if you that would increase dialysis flow will provide better concentration gradient across the membrane, and thus enhance the diffusion. But as you can see simulated in this figure, this is an infinity dialysis flow. So with the practical dialysis flow rate 500, 600, the improvement is very minor. So we also simulate what if we can develop some method that can significantly increase the free fraction. In this simulation, we used a method that could displace solids from albumin, and you can see a significant increase of removal might be achieved by hemodialysis. So since free fraction has so many problem issue to be removed, so what about removing the bound fraction of protein-bound uremic toxin? Well, that means you have to remove the carrier, the protein, the albumin. The cost of directly removing them may not be affordable. As you can see, it's well known that a loss of albumin is not favorable for patient outcomes, and this is maybe not suitable for all patients. And the worst thing is comparing to albumin levels, the level of those toxins are relatively low. So for example, almost every 6 albumin carries 1 induxyl sulfate. So if you really want to remove the induxyl sulfate molecule, you will need to sacrifice a lot more albumin to gain minor improvement for the protein-bound uremic toxin levels. So the loss could be too aggressive to outweigh the increased associated risk with losing those beneficial molecules. So since we can't depend on making the pore size larger to lower the levels of protein-bound uremic toxins, we will have to depend on other possible revenues, such as controlling its production through manipulating microbiota metabolism. There are a lot more studies on this topic than showing here. Most of them showed efficient lowering of toxin levels in the study as an outcome. I only list those who have shown decreased toxin levels and improved patient outcome at the same time. So as you can tell, there are some studies showed a successful story. Some are not so successful, but overall, I think it's quite a promising direction to go to. So to sum up, the evidence-linked uremic toxin elimination concept propose making decisions of which toxins should be removed based on the available evidence of their toxicity. Through analyzing the list of uremic toxins by this concept, we found that the bulk of uremic toxin load are water-soluble small molecules or smaller size metamolecules. They can be adequately dismissed by high flux membrane. Better removal of those difficult to be removed toxins such as protein-bound uremic toxin cannot be achieved by manipulating the pore size alone. So with the concern of increased probability of losing beneficial substance. So I think this concludes my presentation today, and I will be happy to take questions.
Thank you so much, Dr. Tao, for this very clear and interesting presentation on removal of uremic toxins. So it's now time for question and answers. So you should please feel free to submit your comments and questions in the user panel. So until we have some questions, I have some questions for you. And I was intrigued to see your data on TMAO and CMPF. Do you think that these uremic toxins could be used as a surrogate marker for nutritional intake of red meat and fish?
I think that's a good idea. But maybe before I can answer this question better, could you clarify what's the purpose of using them as a surrogate? You mean as a surrogate to evaluate how much toxin generated by the diet?
Yes, so I think that's one of the major concerns in this patient group, especially before they start dialysis, if the composition of the protein intake, if it differs regard to if the patients eat a lot of red meat or have other protein sources such as from fish, which should theoretically be more healthy for these patients than red meat.
Oh, I see. Yeah, I think that could be feasible. But one difficulty that may come to my mind is the individual concentration, the variation between individuals for those toxins varies a lot. The diet content may not be the only factor. So I haven't really followed any study that address this particular point. It's very interesting point to study. But I could imagine a possible result. You see, it's even the patient who are taking the same diet still have very different concentration of those toxins between them. So that may become a difficulty to really use them as a surrogate.
Thank you. Another question here. Water-soluble toxins. Water-soluble toxins are easily removed by current. So water-soluble toxins are easily removed by current hemodialysis technologies, yet their levels are still elevated in end-stage kidney disease. What can we do to solve this problem?
Yeah, so it is true that there are still some water-soluble toxins. They have elevated levels. But it's a good thing that hemodialysis first can bring down their toxin level, the toxin level after dialysis. So if you look at the time exposure of those toxins, water-soluble toxins may have lower exposure comparing to protein-bound uremic toxin whose concentration is constantly high in patients. So that's the benefit of water-soluble toxin being able to remove by hemodialysis easily. But at the same time, there are also toxins, for example, phosphate. They are isolated in a tissue compartment, not easy to mobilize out into the blood compartment to be removed. In those cases, of course, manipulating membrane will not help. We will have to think about strategy like more frequent hemodialysis, longer treatment, and modify the hemodialysis features and modalities to enhance the removal of those toxins.
Thank you. We have a question in the chat, and I read this question now. Is a more dialysis concept, would a more dialysis concept be more important than filter characteristics?
If I understand this question correctly, this question is referring to higher frequency of dialysis, rather than focusing on the pore size, etc. I guess I would say our evidence-based linked toxin elimination concept, it's actually trying to find what's the best one of strategy that to deal with each toxins. So more dialysis could be a strategy that to be identified by analyzing those toxins filterability. I think the phosphate it's an example that could fit into this question. If a filter cannot, if a toxin cannot be simply manipulated or controlled by the filter, then we will have to think about different strategies like more dialysis or modifying the treatment parameters. I hope I hope I answered this question.
Yes, you did. I have another question for you here. What are the technologies that have been tested to enhance the removal of protein-bound uremic toxins in the literature?
There are actually a lot of very interesting and bold and innovative methods has been tested in the literature. Unfortunately, none of them is on the way to commercialization, as far as I know. But one example I can think of is the one I participate in. It's called displacer enhanced hemodialysis. So this method used the binding competition between uremic toxins and other molecules on albumin. So, for example, we can try tryptophan, which is a very minor and even nutritional molecule, to load the albumin before the toxin and albumin go into the dialyzer. So once the free fraction of those toxins is increased, they will be removed through dialysis easily. So this is one of the example, and there are other methods like changing the ion strengths of the dialysate content, because the binding also depends on the ion strength. So if you change the ion strength, there could be more free fraction of those toxins mobilized into dialysate. In this case, you have better removal. But this method may have some hemocompatibility issue, and I think there will be more testing to be done. And I think there are also more mature method like plasma separation method. So once plasma is separate from blood, and they will set into absorptions, those ion exchange absorbers, and the toxins will be ripped off from the albumin. In this case, only the clean albumin returns to human blood. So a similar concept like activated carbon coated membrane that also been proposed, and also absorption added dialysis method that's also been tried. So I can say that there are lots of interest of developing something that could work to remove those toxins.
And alluding to this, do you think that and probiotics could help to reduce the toxins from the gastrointestinal tract?
Yeah, so as I mentioned, there are tons of study, good evidence, also randomized control trials showing that these two prebiotics, probiotics, symbiotics, all kind of therapies has been efficiently reduce the level of especially gut-derived uremic toxins. And I think this is no surprise, as you know, the Japanese medication, the oral Zorban AST-120 has been on market for a long time. It has the similar mechanism of manipulating the microbiota, reducing the precursor. So that also has been shown worked in terms of reducing the blood level. Now, I think the difficulty is, or what we are lacking in this area is to show this whatever therapy that reduce the uremic toxin will really associate or lead to improved patient outcomes. And I think it's also difficult to find the appropriate or the perfect endpoint to be evaluated. So at this point, it's still hard to say what therapy is the magical bullet. But I think overall we have some promising direction to go to.
Thank you. And here is another question from future membrane therapies development perspective. How much additional clearance of protein-bound uremic toxins could we achieve?
Thank you. This is excellent question. I think for protein-bound uremic toxin research, I think we are not at a point we can say we know everything yet to really conclude what how much removal we really need to see a strong improvement of patient outcomes. I think the best evidence we have right now, like I mentioned, the AST-120, which is something that has been studied in many randomized controlled trials. With the regular dosage of 6 grams per day, they're able to reduce plasma level 20 to 30% over time. Without knowing what endpoint we are looking at for improving protein-bound uremic toxin levels, let's say we just want to outperform AST-120, then you will need to achieve pretty much the same efficacy as the therapy by hemodialysis, right? So we did some in-house simulation before. So currently the reduction ratio of hemodialysis for protein-bound uremic toxin, for example, inductyl sulfate is 30 to 40%. And we found that if this reduction ratio can be boosted for another 60%, so you get totally 50 to 60% reduction ratio. Over time, it may have an impact on the plasma level that's similar to what AST-120 can achieve. Now, so AST-120 also, it's not a magical bullet. As you can tell in some clinical trials, they do have some good outcomes in declined reducing the decline of kidney functions, but it doesn't have too much impact on the overall renal outcomes like initiation of hemodialysis, things like that. So, and also many of this is not relevant to hemodialysis patients. So even we get to the same performance as AST-120, we still cannot guarantee a significantly improved outcomes in patients, in hemodialysis patients. So, but it provides a bottom line that we will need to get a reduction ratio more than 60% for inductyl sulfate, precursor sulfate. Otherwise, there will be nothing we can see if we ever run a clinical trial to prove how much additional removal we need for those toxins.
So I would like to thank you, Dr. Tao, for this really excellent talk, and I think you have been very clear about this. It's been very instructive. I learned a lot. I'd also like to thank the audience for having joined us today, and I also want to say that you should be sure to register for our upcoming webinars. We have the next one on June 7th, and then Dr. Stefano Stewart will present, and we also have on June 14, Dr. Madhukar Misra will present. And with this, I say goodbye and hope to see you soon.